Hello, everyone. I am Ana Nieto, Head of Species Conservation Action at IUCN. It is my pleasure to welcome you today to this webinar, which focuses on species conservation action for freshwater species. Freshwater ecosystems are among the most biodiverse on Earth. Despite covering only 1% of the planet's surface, they account for at least 10% of all known species. They also provide critical ecosystem services in water quality, nutrient cycling, and food provisioning, supporting both biodiversity and human livelihoods. Yet, freshwater species are under immense pressure from pollution, habitat degradation, invasive species, overexploitation, and climate change. The recent IUCN Global Red List assessment of freshwater animals revealed that 24% of the world's freshwater species are at high risk of extinction, which poses huge risks for biodiversity, human well-being, and our economy. The IUCN study also emphasized that species like crabs, crayfish, and shrimps are facing even a higher risk, with around 30% of them threatened, highlighting just how urgent the situation has become. This data underscores the urgency for focused, sustained conservation efforts. At IUCN, we are committed to supporting freshwater systems as essential for protecting biodiversity, climatic stability, and driving sustainable development. From freshwater governance and stewardship to conservation action on the ground, protecting and restoring freshwater ecosystem is a priority outlined in the IUCN program, which guides the work of our entire union. Today, we're here to talk about conservation action for freshwater species from IUCN's grant-making programs, particularly the Fondation Segré Conservation Action Fund and the Save Our Species Sturgeons Initiative. The webinar brings together experts and practitioners across the whole world, from Africa, Asia, Europe, and Latin America, that will showcase conservation results, share lessons learned, and explore innovative solutions to tackling drivers of species decline. Their efforts ranging from river restoration and invasive species control to community engagement, genetic conservation, and habitat monitoring demonstrate the power of collective action in reversing biodiversity loss. With that, I would like to introduce our facilitator today, Dr. Topis Contreras McBeath, who will be guiding us through today. Topis is a long-term friend of IUCN and the Species Survival Commission, and he co-chairs the IUCN Species Survival Commission Freshwater Conservation Committee. I hand over to you, Topis.
Gracias, Ana. Well, greeting from Mexico. It's really nice to be here moderating this incredible panel with what Ana has mentioned about the freshwater biodiversity crisis. The question now is, how do we reverse this crisis? What do we need to do to go beyond assessments? Most of the time at IUCN's Species Survival Commission, we have been focusing on assessments. We recently finished the Global Freshwater Biodiversity Assessment, as Anna mentioned, and we found that a quarter of freshwater species are threatened. But we want to go beyond assessing. For the past maybe 8 years, the SSC has implemented what is called the Species Conservation Cycle, which seeks to, from the assessment, doing conservation action plans and then moving towards conservation actions to protect freshwater species. And in the past maybe 8 years, a lot of impulse has been given to the conservation of freshwater species. We used to be having a great difficulty in getting funding for this endeavor. But fortunately, we now have foundations like Segré, which are giving money for freshwater species conservation, and also the funding from IUCN is also giving. And so today, we have put together a really interesting panel of presenters that will be talking about some projects from all around the world, different contexts. We have Dr. Jeremy Shelton, who's going to talk to us about a group of fish he's protecting in South Africa. I won't get into details because he's going to explain what he's been doing recently. Then Diki Juwanto from Indonesia is going to talk about the conservation of a freshwater shrimp species. You know, we also don't need to focus only on freshwater fishes. There's a lot of species that are threatened. We have mollusks, we have fungi, and many other invertebrate species that need our protection. And Dicky will talk about this. And then Thomas Friedrich is going to talk about a really important species of from Europe. So different contexts, different types of focus of how do we do conservation in areas, very remote areas or areas like in the Rhine River, you know, where you have a high concentration of people and industry. And then, of course, we have my very good friend, Mike Balzer, who's going to talk about the 1,000 species program that SHOAL has been implementing in the recent years. And so this will, you know, like sum up of what we need to do in the conservation world related to freshwater species. So the way we're going to work is each of the panelists is going to give a 10-minute presentation. And after that, we're going to have a panel discussion for about 20 minutes. And the last part of this discussion, all of you from the audience will be able to submit questions, and we'll send it around into our panelists so they can answer. So without further comments, I present to you Dr. Jeremy Shelton, who will give us his talk. Go ahead, Jeremy. We can't hear you, Jeremy. Something.
Can you hear me now?
Yes, we can. You can start.
Sorry about that. Cool. Yep. Great to be here. Looking forward to a really diverse and interesting session. Let's go for it. Today, I'm going to be telling you about the fynbos fish revival in South Africa. I'm based at the Freshwater Research Centre in Cape Town, and I'm a research associate at the South African Institute for Aquatic Biodiversity. And yep, at the core of this talk is going to be some of the work we've been doing recently with support from SOS, IUCN SOS, and Fondation Segré. Next slide. So over the last 50 years, the size of monitored wildlife populations across our entire planet has declined pretty sharply by about 73. I think it's now close to 75%. But the decline in freshwaters has been measurably sharper than in marine and terrestrial systems. Next slide. And this is a pattern that repeats itself here in South Africa. Our National Biodiversity Institute does a biodiversity stocktake every 5 or so years. And these are some of the results from our most recent biodiversity assessment. And the top panel shows the threat status of our ecosystems and our species groups. And what jumps out here is that our freshwater systems, the rivers and the wetlands, really stand out as the most threatened ecosystems in South Africa. And it's the freshwater fishes that are the most threatened species group. And then the bottom panel shows basically an inverse of these trends. If we look at protection level, it's these rivers and wetlands that are the least protected, and the freshwater fishes, again, that have received the least protection. Next slide. And the reasons behind this are complex and interacting. We have human-linked impacts on water quality and water quantity, all of which have been compounded by the effects of climate change. And then invasive species in our river basins have caused huge declines in river flows and the smothering of aquatic habitats. But probably the biggest threat to our indigenous fish species is that of introduced sport fish like the predatory bass and others from the Northern Hemisphere. Next slide. And all this is really the backdrop for some of the work we've been doing, which has been focused on the southwestern corner of South Africa. It's a region that's best known for its exceptional plant uniqueness and diversity, nearly 10,000 species of plants, most of them unique to the area. But what's less well known is the very high levels of endemism and threat among the freshwater fishes beneath the surface of the rivers, flowing through this biodiversity hotspot called the Cape Floristic region. We've got a very high concentration of unique and threatened species, although far less diverse than the plants. Next slide. And that's really the backdrop for launching the Fynbos Fish Revival, which is a mission to revive the most threatened freshwater fish species in the Western Cape corner of South Africa through critical fish habitat. We are going to improve the conservation status of the 10 most threatened fish species in this region over the next 10 years. And we're going to do this through collaborating with a range of different stakeholders. But we've been particularly working closely with marginalized communities where unemployment's high. And we believe there's great potential for realizing the dual goals of creating jobs and restoring freshwater ecosystems at the same time. We're also really partnering with other NGOs, government, and landowners, and trying our best to connect globally to align with wider initiatives and strategies as well. Next slide. We use a mix of different kind of approaches depending on the situation. We work to increase the footprint of protected areas, to restore critical habitats, species reintroduction programs, managing water resources at a catchment or a basin scale. We're committed to science-based conservation action and adaptive management, and we have a strong emphasis on visual storytelling to build support for this work among different stakeholders. Next slide. And yeah, I'm really proud to share with you all some of the highlights from our last 5 years of work on the Fynbos Fish Revival Initiative. So I'm really presenting on behalf of a range of different people and organizations. On the biodiversity side, we've restored 22 kilometers of river. That's now up to about 30 and counting. We're doing this primarily through invasive species removal, both underwater and above water, and also through working with landowners to balance water flows. We've assisted 9 endemic fish species in the region. And then on the community side, we've created jobs for 12 people. And that's converted into a lot of days of employment. And we've also connected with a growing number of landowners and youth groups as well, connecting them all to the conservation work in the area. Next slide. This shows the spread of some of our projects that are active at the moment or recently completed. And it's really starting close to Cape Town and spreading up north through the region. Close to home in Cape Town, we're working on reintroducing an endangered species of fish called the vitfish to a river system where it's disappeared 30 years ago. And it's really exciting time for that species in that system. And then all the way through up to the north where we've been working on the Clan William sandfish for the last 7 years. Next slide. The Bedo River is a critical habitat for the Clan William sandfish. It's South Africa's most threatened migratory fish species, and this is a location of an ongoing reintroduction program where we've been rearing and releasing sandfish to rebuild a very, very depleted population in one of the last spawning tributaries for this species. And then building on that, we've recently undertaken an ambitious project backed by the IUCN Save Our Species and Fondation Segré, which is looking at habitat restoration to really secure the sandfish in the long term, but also to use the sandfish as an umbrella for other fish species, other freshwater species, in fact, the whole ecosystem. And the idea is to generate benefits for downstream landowners as well. Next slide. So this river has been invaded by North American predatory fish in over 90% of its length, and the indigenous species are pretty much pushed into a corner right at the top of the catchment, a very short section where they're protected from invasion by a waterfall. And this is a pattern that repeats itself all across the Cape, all across South Africa, and no doubt elsewhere in the world. Next slide. So our plan was to eradicate the invasive fish species from the river and to extend the range of these indigenous fishes from 1 kilometer to 10 kilometers. And the question of how do we remove invasive fish from rivers is a very complex and interesting one. In some cases, we can do mechanical removals, but in this case, due to the nature of the river ecosystem, the habitat complexity, and the species present, we had to do a chemical alien fish eradication. And this is really building on work done a decade ago by the South African Institute for Aquatic Biodiversity, Cape Nature, our local conservation authority. And we used rotenone, which binds to the gills of aquatic species and stops them from being able to absorb oxygen. Next slide. And then at the downstream end of the restoration zone, we invested in a barrier weir to prevent the invasive species from coming back in the long term. And here you can also see the neutralization of the rotenone at the end of the treatment with potassium permanganate. Next slide. This is just a snapshot of what it looks like to do an alien fish removal with rotenone in this part of the world. We had a big challenge with a very difficult to access catchment that we had to open up. We used control burns and aerial spraying to access inaccessible areas. We had to use backpacks to spray side channels and backwaters, in addition to our drip stations, sentinel fish to make sure we got the concentration just right, and a parallel invasive plant removal project, which has its own story, which is for another day. Next slide. As we wind down, I can report from the field a couple of days ago that our early data are showing that it looks like it's been successful. We found no bass and bluegill through those 10 kilometers, and we're starting to see recovery of the indigenous species through that zone, which for me is just the most inspiring thing. And it's a really great start, but there's still a lot to be done. Next slide. And I really want to acknowledge all the different individuals and organizations that collaborated to make this project possible. It's definitely not a one-man show at all. Next slide. And my last two slides, some takeaways for this webinar. I think this is a very exciting time in history for freshwater conservation. This is where we get to bend back the curve of freshwater species loss. The challenges are large, and the solutions need to match that in largeness. Business as usual ain't going to cut it. I think we need to be bold and experimental, but also strategic, data-driven. We need to be agile and ready to pivot and adapt. And my last point for my talk is, how do we scale up these conservation wins where resources are limited? And I believe that collaboration and marketing are two of the most important ingredients in this. By working as collectives and communities and consortia rather independently, there are huge benefits for increasing our impact, our sustainability, our knowledge sharing, and our fundraising. But then on marketing, scaling up our conservation wins requires substantial investment, and that's something that we biologists are notoriously bad at. So it's wonderful to see publications like The 1,000 Fishes, the Africa's Forgotten Fishes, really raising the profile of these species and these ecosystems and really helping to channel investments. And slide change, please. My last slide shows an example of how these things can all work together to hopefully maximize our impact. In this case, we've got a global strategy. SHOAL created this list of the 1,000 fishes are all around the planet, most in need of assistance. And then we've had strategic investment from backers like Rewild, who are allowing us to channel resources into local impact where we're going to succeed in conserving our most threatened species in this area over the next 10 years. Thank you very much.
Thank you very much, Jeremy. And we turn now to Dicky.
Yeah, thank you very much, Tobis. Can you hear my voice?
Yes.
Yeah. Well, hello everyone. Good night from Indonesia. My name is Diki Dwianto. Now I'm pursuing my PhD degree at Animal Bioscience Study Program at IPB University. Now I'm also acting as a head of Masyarakat Zoologi Walase, a local NGO based in Sulawesi, with our focusing on conservation of various endemic species in Walase. So it's such a great honor for me to present my project in Lake Poso about the conservation of endemic freshwater shrimp, genus Caridina. This project was supported by Fondation Segré through the IUCN Save Our Species under the Research Support Grant Scheme. Next, please. So I will begin my presentation with the background of this project. So we are now coming to Sulawesi Island, the only island in Indonesia that harbors two ancient lakes, Lake Poso and the Malili Lake system. Lake Poso is located in the central highland of Sulawesi. This lake was formed for at least 3 to 1 million years ago. This lake contains rich endemic freshwater species. So this lake has been well known as a natural laboratory for many researchers. So several species flocks can be found in this lake, including endemic fishes, crabs, snails, and shrimps, especially genus Caridina, with a total of 11 species described up to the present. However, only 6 species have been assessed and classified as vulnerable based on the assessment in 2011. It's about 14 years ago. And the main target of this project were the 6 species, including Caridina caerulea, Caridina insifera, Caridina acotrostris, Caridina longidigita, Caridina soricinorum, and Caridina senkeli. However, during our study, we also recorded other species that have not been included in the IUCN Red List database for their population assessment. And as we know, Caridina from Lake Poso also suffers from various ongoing threats, including habitat destruction and land conversion along the lake shore, pollutant input from agricultural and residential areas, overexploitation because some species are sold by the local fishermen as a fishing bait, and of course the invasive species. Next, please. So the main purpose of this project is to gather scientific evidence that will facilitate the conservation of Caridina species in Lake Poso. We started our project in January 2024. We combined multiple methods to collect the data on the population and of freshwater shrimps, including hand net, while snorkeling, tri net, deep net, and also scuba diving in the deeper part of the lake. So next, please. So, well, yeah, in this project, we successfully implemented 5 key conservation actions to provide scientific evidence to devise critical conservation action in the future to make these tiny crustacean creatures, Caridina shrimps, safe from extinction, and allow the local community to appreciate the many of endemic species in Lake Poso. So the first action was to conduct population assessment for all of Caridina species by using cats per unit effort methods in 33 sites surrounding the lake. This was the first comprehensively sampling for relative abundance of Caridina in Lake Poso, including their habitat preferences and their ecology. The second action was to determine what is ongoing threats in the habitat of Caridina. This data will be crucial to identify what is the best action to reduce the impact on the shrimp population. And the third action was to provide distribution map of Caridina based on the present absent data collected in the 33 sites, and to identify the area that potentially develop as a key conservation sites. And the fourth actions was to increase the local community awareness by introducing Caridina in Lake Poso to the young generation and linking the scientific information to the local community. So I think that the research findings should not only be used by scientific or students or lecturer, but also can benefit local community to increase their sense of belonging to their diversity of endemic species in that area. And the last action was to increase public participation by using science citizens platforms such as iNaturalis, where many people can observe the diversity of Caridina in Lake Poso and uploaded the data to the project database. Next, please. So the first result for our project, we successfully recorded the relative abundance of 10 out of 11 species of Caridina in Lake Poso, and among them Caridina ensifera and Caridina longidigita were the most abundant species in the lake, in contrast to endemic species that have not been assessed so far. Caridina poso and Caridina fusca showed the lowest relative abundance. And the abundance of Caridina was correlated with their habitat preferences, with species that have high population abundance belong to the generalist species, or they can be found in the various type of habitats. And unfortunately, we cannot find one species, Caridina lilianae. The species just described in 2021, but the species now is disappeared from the two localities where the species was found for the first time, and we will continue to looking for the species in the future. In the second result, we found several threats in the habitat of Caridina in Lake Poso, including invasive species, overexploitation, habitat destruction, and habitat loss. Recreational activities surround the lake and potentially pollutant input from the rice field area in Lake Poso. The analysis of human disturbance level showed that only several areas habitat for Caridina in Lake Poso received environmental pressure, as you can see in the map with the red color. The sites mostly located both near the human settlement and in the agricultural area. In the third result, so this is the first, the distribution map for Caridina, all of the Caridina species in Lake Poso. And as you can see in here, there are some species that are widely distributed in the lake, and several species have a specific distribution. Caridina ensifera was the most widespread species and could be found in almost all sites. And the fourth result, this is especially for the education program. We conducted seminar in two senior high school and one university in Lake Poso, with a total participation of about 100 people, including students and teachers involved in this program. We also introduced about how to use iNaturalis software to the student from the university, and taught them how to make an observation and uploaded the data to the project in iNaturalis. Next, please. So this is the additional program. So far, there are 64 observation in iNaturalis project, and we also published several blog and news in the local news, and also we conducted mini museum in the schools in Lake Poso to introduce the diversity of endemic species to the students and also young generation. Next, please. So the challenges and lesson learned from this project. So for the main challenges, I think after we know that the several species of Caridina have a very low abundance in the lake, and the problem from the invasive species, especially for species that not used by the local fishermen as that have economic importance, I think involve management agency or government that have resources is the main challenges, because we need big resources, big effort to eradicate the invasive species given by the large area of Lake Poso. And the second challenge is to link with the international expert and IUCN representative to updating the status. Yep. Of Caridina. And the last challenge is to bring together all of the local NGOs into one big action to conserve endemic species. So lesson learned, we were able to working with together with the local NGO and community, and I think we also need to provide establish a new way to communicate scientific data with local community, and collaboration is a key, including the scientists, hobbyists, conservationists, lecturers, students, local people, and also government. Next. So we are grateful for the support from various organizations and also research institution and university. And this project would not be possible without support from Fondation, Segré, and IUCN Save Our Species. Thank you.
Thank you very much. We turn now to Thomas.
Thanks, everybody. I'm Thomas. I'm a scientist at Oak University based in Vienna, Austria, and also active in the World Sturgeon Conservation Society, Danube Sturgeon Task Force, and IUCN Sturgeon Specialist Group. Next, please. So I'm going to present you the SOS Sturgeon Grant Project Sturgex. Maybe before I start, so sturgeons are an ancient group of fish, nearly 200 million years old. We have around 27 species distributed along the northern hemisphere, anadromous species, bottomodromous species, and populations of both forms. And sturgeons are generally interesting because they can grow quite old, up to 200 years, and some species can reach up to 6-7 meters in length. The last IUCN Red List assessment showed that in 2019, when it was done, that we actually deal with one of the most threatened families of animals here. Next. So the region we're working at is the Danube River Basin and the Black Sea. It's a very challenging area to work once we are in the most international river basin globally. We have just in the sturgeon range 12 countries along the Danube, plus 3 more countries along the Black Sea coast. You're, I think, pretty well aware of the geopolitical challenges that we have to face in the area. And of course, there's very different socioeconomic and political systems and situations in play. Reasons why these fish vanished, and that's more or less the same in all parts of the world, is for once the overfishing for the meat and the caviar, then blocking of the migration routes, damming the rivers. And in the Danube River, what we are facing is an ongoing threat through poaching. So there's projects also through this IUCN grant where this threat is approached. And but we are in the fortunate situation that we still have more than 800 kilometers between the Black Sea and the first migration barriers. Next. So when it comes to the conservation, we need a two-pronged approach. We need in situ conservation, meaning protection of the remaining individuals in the wild, protection and restoration of habitat and migration routes. But unfortunately, once populations are depleted in a way that they might not be able to revive themselves, we also have to deal with so-called ex situ conservation, meaning we have to keep the species in captivity and try to reintroduce or stabilize the populations in the wild by the releases of offspring. And I will now mostly deal with this ex situ part. Next. So the basis for an ex situ propagation program is the maternal animals, so the broodstock. These fish need to have a proven genetic origin. They should be genetically diverse. You should be able to integrate mating schemes based on their relation, and you need suitable infrastructure with little risks, fail-safe mechanisms in order to maintain these animals. And next, for that, this IUCN project works together with a live project, Lifeboat for Sturgeon projects that we're implementing in parallel. And you can see here the facility, which is a floating rivering facility that was built out of a 60-year-old ship that's running with river water to maintain the broodstock as well as to rear the juveniles. Next. So the way these projects work together is that the LIFE project provides infrastructure, manpower, public relations activities, monitoring work packages, while the IUCN SOS project provided us with the ability to go out, look for fish in captivity in caviar farms, screen those for their origin, if they're of Danubian origin, investigate their relation, and then obtain mature broodstock to be kept in the boat in our facilities to artificially reproduce and then rear the offspring, and then release the offspring out into the wild. Next. So these are just a few numbers, but really what the goal was of this IUCN project is to enable us to purchase broodstock of the beluga sturgeon, Russian sturgeon, and stellate sturgeon, and all of those, of course, based on their origin and the diversity that we encountered in various farms all over Europe. Next. The way we're dealing with the offspring is that we incubate and rear them in river water. We want those fish to exhibit the so-called homing behavior, to be imprinted on the river. We try to use methods where we mostly rely on live and frozen feeds that they would encounter in the wild as well. And we have targeted release strategies when it comes to the size, when it comes to the location, and when it comes to tagging and genetic variation of the offspring. Next. So one thing that I'd like to highlight here is the release of the Russian sturgeon. So just this year, we were able to release 120,000 juvenile Russian sturgeon, and those are in fact offspring of the broodstock that we were able to obtain through the IUCN grant. Next. Yeah, without spreading the enthusiasm, there is no funds for conservation. So a major point for us is to engage the public, to engage stakeholders, to engage our students, to engage school kids, to be creative in this regard. And this really pushes the whole topic. We are in the fortunate situation that we have a sexy species, but this really pushes the whole issue of conservation forward. Next. Yeah, so especially in species that take a long time to mature, like sturgeons, we have to look way forward into the future, and we have to develop strategies how to maintain activities into the future. And this can only happen when multiple stakeholders work together, when multiple projects work together, and when we can really engage with other entities like the public and other potential stakeholders. And just for you as an example, what we're doing, for example, is that our broodstock is up for adoption, and single individuals, companies, whoever can actually adopt this broodstock and hence provide a baseline funding for maintenance of this broodstock well into the future. Next. Yeah, what are things to take away from the whole topic? So when you implement ex situ activities, so especially for sturgeons, we published many scientists from all over the globe published these guidelines that you can see here. But the baseline is when you do ex situ, you cannot jeopardize the genetic integrity of a residual population. If you have one, you should be able to separate wild recruits from stocked animals to see how the population develops. You should be aware that a small broodstock can lead to potential genetic bottlenecks. With regard to your releases, you should try to avoid the genetic dominance of individual families, and you should promote phenotypic diversity, fitness, and homing behavior in the fish you release, and avoid any domestication effect. And especially when you're dealing with a catchment like the Danube, like the Baltic Sea, like the North Sea, where there's multiple countries, multiple institutions, coordination is key. It's very tricky, it's really time consuming, but you have to coordinate with all those involved in both positive aspects as well as negative aspects, such as hydropower development or fisheries. And this is something that is really tricky to circumnavigate when it comes to the acquisition of funding, and the political interest, of course, is that you cannot guarantee results within short time frames. We took nearly 200 years to exterminate the species. We cannot expect to bring them back within 5 years. So this is something that should be within your communication, that patience is for sure required. Next. Yeah, and with that, I'd like to thank you for the attention. The sturgeon world is a pretty small family who are all very enthusiastic about the conservation and preservation of these magnificent old fish. So whenever you're interested, reach out to anybody who's working on the topic. Thank you.
Thank you, Thomas. Okay, it's your turn, Mike.
Thanks very much, Thomas, and great, wonderful presentation so far. It's good to hear them all, and fortunately, I know very well, and the work that they're doing was fantastic. And particularly Jeremy, just a shout out to Jeremy, who's not only a fantastic fish expert, but an amazing photographer, and you're likely to see a few of his photographs in my presentation. So let's move to the first slide. Thanks very much. So again, just re-emphasizing how important, can we go to the next slide, sorry? Okay, so re-emphasizing how important freshwater biodiversity is. It's really the lifeblood of the entire planet, and pretty much all the biodiversity that's non-marine is really dependent on these freshwater habitats and the intact freshwater environments as well. So, and just a thought about what would we lose without these intact freshwater habitats, and particularly one side of it I'd like to mention about is fisheries. Next slide. So one of the things that most people don't appreciate is that actually inland fisheries is pretty much similar to the size and scale of marine fisheries. And very recently, I was at a meeting where a fisheries expert was suggesting that inland fisheries, in terms of food security, is on the same level as agricultural, same level as agriculture globally. So these freshwater ecosystems are not only important for biodiversity, but important for feeding and supporting some of the poorest families and communities across the globe. Next slide, please. And so that there's an enormous number of people also involved in this industry. And next slide, please. And more than 50% of those people are women, which is it sounds like normal, 50%, but actually this is very high for an agricultural sector or for any sector that so many women are involved in it. So it's a critical point of employment and food security for millions and millions of people. So the decline in fisheries, decline in freshwater habitats is something we should all be concerned about. Next slide, please. And as I mentioned earlier, IUCN produced a landmark paper summarizing the work of thousands of scientists over the last decades and understanding where we are with freshwater species and freshwater habitats. And as Anna mentioned, 24% of those species were recognized to be threatened. And for freshwater fishes, this is as much as 26% and maybe higher, as you'll see when we talk a bit later. So we have this paper was very important to establish this understanding that really there is a freshwater species crisis. Next slide, please. And we've seen for various statistics in terms of the loss of the wetlands, 35% of lost wetlands in the last 50 years, 80 freshwater species lost, and it's twice as many of the freshwater species have been lost than marine or terrestrial, and that's all happening in just 1% of the Earth's surface. So it really is a concentrated effort on a very important set of biodiversity and a set of habitats. Next slide. If we just take fish diversity, for instance, there's roughly, when we did this, it was about 18,000, but it goes up every year, roughly around 250 species. So there's an enormous number of freshwater fishes, and if you compare that to the total number of birds in the world, something like 11,000, you can see what incredible diversity there is. And with 3,000 or more of those are threatened, and a very large number of them, over 2,500 of them, are data deficient. So, when you add those two together, and you assume that some of these data deficient species, or all those data deficient, are to some extent threatened too, then we're really talking about a third of all freshwater fishes are threatened with extinction. Next slide, please. And this is matched with a situation where freshwater species conservation, freshwater in general, has been very neglected and underfunded, and this is part of the underlying cause of this crisis, that there's considerably less investment in terms of funding, in terms of organizations, in terms of research, etc., compared to marine and terrestrial environments. Next slide, please. And it was in response to this situation that a number of experts and organizations came together in 2018 and said, okay, well, how do we deal with this situation? How do we end this freshwater crisis? And from that consultation and those workshops, SHOAL, an organization, an initiative called SHOAL, came out of that, which I'm very fortunate to head. Next slide, please. So SHOAL was basically the mission of SHOAL is to end all freshwater species extinctions, and it's very, very focused on targeting and direct action for freshwater species, particularly freshwater fishes. And it was also there to complement the plan, the strategy of the IUCN SSE of plan, assess, plan and act. And SHOAL is very much coming in from that act point of view. So when we looked at when SHOAL was set up, we looked at some of those major issues facing freshwater species, and we came up with our strategic pillars, and we call those act, inspire, and mobilize. And ACT is responding directly to the rapid loss on the ground of freshwater species and habitats, while INSPIRE is responding to the fact that really there's a general lack of understanding and awareness of the importance of freshwater species, and even to the level where we really don't understand and love freshwater species as much as we do other sort of terrestrial species that have had a lot of attention in the media and films, and etc. And fundamentally, one of the biggest problems is that we really have a lack of funding and knowledge and capacity across the globe to address this the freshwater crisis. And these are the three precise areas that SHOAL was developed to focus on, and particularly with ACT. If you think about SHOAL, just think about the word ACT, because everything this initiative does is to get action on the ground. And some of the ones we've just been heard from Dickie and Thomas and Jeremy are really exactly the sort of things we want to see more and more of. Next slide, please. The way we set up SHOAL is essentially it is a partnership. We're totally inclusive, and we have two types of partners. One we call strategic partners, and one we call the local action partners. And so the strategic partners are those organisations, zoos, universities, museums, donors, all with some capacity to really help support and generate and escalate action for freshwater species. And then what we're doing in SHOAL is trying to target that and focus that capacity to local action partners. Those are the people on the ground really doing the great conservation action, sort of actions that Jeremy was talking about in South Africa. So that by creating this collaboration and capacity with strategic partners, we need to really begin to support those local action partners, but really make a difference. And so we've, in the last few years since SHOAL was launched, we've got about over 35 strategic partners, 30 local action partners working in 5 continents. We've created 2 alliances, one of them being an aquarium between an alliance between zoos and aquariums that are playing an extraordinarily important role in freshwater species conservation, particularly freshwater fish conservation. And we're targeted over 200 species already since it was established in 2019. Next slide. 2 years ago, we started an initiative called 1,000 Fishes, and the goal of it is to get conservation action for 1,000 of the world's most threatened fishes by 2035. So the whole goal and the aim of this initiative is to really escalate the quantity of actions going on for different fishes, and in turn create more and more capacity, mobilize more and more funds, and basically try and catch up to bend that curve towards of action and try and catch up with the other groups, the terrestrial marine species. Next slide, please. So as part of the 1,000, the first part of the 1,000 Fishes initiative was to do an analysis of the situation for all these priority fishes and do a sort of deep understanding. This also came at a great timing in terms of when the IUCN released their paper on the freshwater assessment. And so we were able to start looking at patterns and understanding where these fishes are, what are the common threats, what countries have where are these fishes concentrated, so that we can focus our attention on that and looking at those places that have the least amount of capacity. And so it's also, if you go online and look at this, the blueprint, it's more or less a business plan in terms of how the freshwater fish conservation community have targeted and hope to target these fishes to get to the point where we're achieving this goal of 1,000 fishes by 2035. Next slide, please.
You have 1 minute, Mike.
Oh, okay. So next. Yeah, so we have about 2,500 priority species, and at the moment only about 12% of those are receiving action. So again, we emphasize the idea is really to escalate and accelerate the action for these species. Next slide. So this is just a blatant no excuses, just to show you how wonderful some of these fishes are. But we don't have time to dwell on that, but we'll move on. And one of the main outputs from the blueprint was identifying 36 priority countries where there's a disproportionate number of threatened fishes that need our focus and our attention, but also where there's often a lack of capacity to deal with that. So it doesn't mean we're not working or supporting conservation action in other countries, but these are the places that we really, if we make a difference in these countries, we're going to completely shift and change the situation for freshwater fishes. Next slide. And we've done the analysis is looking at individual countries, for instance, in Indonesia, where there's incredible diversity of fishes, but also an incredible number of priority fishes that we need attention on. So we can target and work with specific countries, and I've got pleasure to say that we work with Dicky quite a lot on freshwater fishes too. Next slide, please. And now ultimately it's all about action and working with the local organisations, local scientists, local governments. At the moment, there's more than 80 organisations working on these 1,000 fishes that we're working with across the globe, and we just need to go more and more. And there's two examples before I finish of that. One is in Indonesia, working on this betta burdigala. It's a critically endangered fish. It's now only found in 15 hectares of peat swamp forest, a tiny area that's highly threatened, and we're able to work with the local organisations, local universities, and particularly Erlangen University, and support them with very little resources to protect this critically endangered species and save it just before it goes. And another example of that is the next slide, please. The red-tailed shark minnow in India. That if anybody, sorry, next slide. Yeah, if anybody has ever kept fish will probably know this fish. Although it's common, I can find one locally down in my local aquarium shop. It's actually critically endangered in the wild, and we're supporting action through Zoo Outreach as our local partner in India to protect the species. Last slide. So my plea for everybody on this wonderful meeting is that if you're working on freshwater fish conservation or freshwater species conservation, please contact us. Let us know what you're doing. If you aren't working on them but you want to work on them, please also contact us, and just to and so that we can really start to enroll more and more organisations targeting freshwater fishes and try and achieve this target of 1,000 fishes. So please contact us and join the show. Many thanks.
Thank you very much, Mike. And with this, we end the first part of our webinar. And now we're going to the panel discussion. Before that, I want to remind our attendees that they can send questions that will be by our panelists at the end of this panel. You can write it down in the chat. We have already received some questions. I'll read them to you later. Okay, but first, I'm going to start with a question for all panelists. So what do you see as the biggest barriers to reversing freshwater species declines, and how can these be addressed through collaboration? Does anybody want to answer this? Any of the panelists? Come on, you have all talked about this in one way or another.
I would say it's pretty hard to define on a global scale. But then, of course, the human development is the major issue to address here. And I think the only way to really go out and change things for the better is to engage directly with people and stakeholders. For us, at least in the northern industrialized regions, hydropower for sure is the major challenge that we have to address. And marine fisheries, when it comes to anadromous species, is the main pressure to address for most species. There are multiple others, but I think those are the major tasks that we have to tackle.
Thank you very much. Jeremy.
Yeah, I think one of the big challenges that we're facing in Africa is just the resources and the support to do the work that's needed. In a lot of cases, we know what needs to be done and we know how to do it, but we really struggle with limited capacity and limited resources to restore critical ecosystems. And I think in my own experience, I found it really challenging working with short-term grants, relatively small grants, and I think we really need to ratchet up the investment, both in terms of the quantity, but also the longevity of support. And I think that's where these relationships, this community, the connectedness that SHOAL's bringing about has the potential to change that funding culture into a deeper investment in what really works. And like Thomas mentioned, even 5 years is just getting started for some of these species that have been knocked so hard. So we need to find ways to get more strategic with our capacity and our support. Yeah, thanks.
Thank you, Jeremy. Michael?
Yes, so just to add on to both of those, I think that that one of the positive sides of what we found from the blueprint and the work that we're doing is that many species don't need so many resources as some other species, and many other very confined to small areas like the Beta Berdegala in Indonesia, where really needs a relatively small amount if you compare it to some other species in terms of investment and capacity. Is providing, and so the ability to get technical support from our strategic partners and some funding, but again, and I totally agree with Jeremy, also of having worked on many of these things myself, is that long-term funding is really critical. So although things can be achieved, quite dramatic changes for some of the fish, not some of the sturgeons, are very, very complicated. But some of these species, if you can find small amounts of funding, but also provide that support to that local organisation and the communities that are involved in the actual direct conservation over time, it's not something you solve overnight with a small grant very often. But the small grants can make a difference. If you compare it to many other types of species, it is very, very powerful for local communities and local organisations, but with the understanding that some things don't change overnight always. So that longer term funding is really, really critical.
And I think another big issue is getting the local communities involved, no? Because they live there, and it's trying to change the perspective of freshwater. Because you can't have funding forever, but if the local community gets involved in conserving the natural species, then that can help a lot. Okay, I'm going to go to the next question. This is only for the grantees. How have your projects contributed to tangible improvements for species habitats? What support mechanisms, technical, financial, partnerships prove most valuable? Dicky.
Yeah, for many years, I think all of the described species of endemic Caridina in Lake Post have had no information regarding their population trend. As you can see in the IUCN database, where there is no population available, and 6 species have been assessed for more than a decade ago. Thus, we need to be reevaluated, including their threats. So this project provides the first baseline data for all of species of Caridina in Lake Poso, and the monitoring program in the future will benefit from the population assessment in this project. And for species management, we identified the conservation priority area that can be designated as a protected habitat for shrimps under the local regulations. In addition, our data on the species-specific habitat relationship show that each species has specific habitat. For example, Caridina longidigita and Sarcinorum strongly depends on the presence of macrophytes and submerged roots in the littoral area of Lake Poso, which is now mostly cleared for agricultural and residential purposes. So I think in this project, we successfully involved local youth and students also to actively engage in conservation of freshwater endemic species through school campaign and seminar. This highlights the importance of partnership, the local organizations, schools, and this could be done without financial support from funders such as Fondation Segré and IUCN Save Our Species.
Thank you. I think, yeah, it's really important to have the best scientific information available, not to see how the trends in conservation. Anybody else of the grantees want to?
Yeah, sure. I mean,
Thank
this
you.
is a very special case, but in our case, so the species was present in private farms, but the access to these fish and their genetic information more or less depended on the goodwill of those farmers, which in many cases was fine, but still in the long term, there's big risk for the species. So for us, what this project provided us was the ability to build a captive or living gene bank of the species that's now more or less public, that can be used by everybody for conservation, and that's like a resource for the next decades. So this is really a big step for us in the whole region. And with the network through IUCN, through WSCS, through all those people engaged, even outside of the Danube catchment, this is really what's helped us establishing the whole program.
Yeah, great. Jeremy?
Yeah, I mean, from my side, I think some of the tangible improvements we've seen is that to actually reverse species declines and restore critical habitats is not that hard if you have the scientific data, if you really understand the system. That takes a long time. So I'd agree with Mike, some of these smaller catalytic grants have a really vital role to play. And then beyond those actions, I think that's where we've found the visual storytelling has been really helpful for raising awareness and connecting different stakeholders around these projects, especially landowners, where a lot of our threatened species are on private land, and so without the investment and the support of those landowners, it's a non-starter. But especially through visuals, signage, it doesn't have to be fancy. It can be like a short film that's shared with the network, and it can go such a long way into opening doors and just getting the support of people in a region to really continue the I guess the sustainability of these projects after they've finished. So that's one of the mechanisms that we've found to be really impactful.
Yes, of course. Having information and then being able to traduce this scientific information so the public can understand and engage. And I think that's very important. Okay, this next question is for you, Mike. How does SHOAL foster partnerships and elevate freshwater conservation at the global stage? And where do you see the biggest gaps in action or investment?
Yeah, so I mean, one of our roles in SHOAL is to profile freshwater fish conservation as much as we can. So getting the stories from the field in terms of successes or some of the priorities for action, and getting those and positioning them within some of these global conversations about priorities is one of our roles. And what was the second part of the question? Oh, yeah, where do we start with the biggest gaps? So the biggest gap is funding. Simple as that, because everything to a large extent falls from that. So there's very few organisations on the ground that it's very challenging to find a single organisation that really focuses on freshwater fish, or even sort of overlooked and neglected freshwater types of biology. And there's a question here about aquatic fungi, for instance. So and that's partly to do to funding, due to the problem of funding, because if you're going to set up a local organisation or a national organisation working on these freshwater species, you really need to be confident there's going to be enough funding around, which until now that there hasn't been. And so we really need to do something about that, and try and ensure that there's more funding. And so the biggest gaps again are really people on the ground that are able to do the conservation work directly on the ground, because and trying to get the support that they need, whether it's technical or financial or anything that they need to really sustain and make sure that their work is impactful. So I think funding is one of the main messages. The second message is, and I say a third one, I would say one of the biggest gaps is finding a way to link the priorities and the conservation actions with more, let's say, mainstream conservation, like protected areas or sort of terrestrial conservation, where there's already quite a bit of funding. So there may be a case where a protected area is getting well funded for another type of species, but it's actually neglecting the freshwater species, and it wouldn't take very much to link those two together. And one of the studies that we're looking for funding at the moment is to try and find the overlap between protected areas and these priority fishes that we have to really sort of then engage with it, where possible, with these protected areas to support them in terms of focusing on the fishes too, but rather than some of the other reasons why the protected area might have been set up.
Yeah, okay. Thank you very much, Mike. Yeah, and SHOAL is playing a big role now in trying to promote this, everywhere. And we thank you for that. You've done a great job since you've been in charge of SHOAL. I have a last question. A question for all of you. What one action or partnership would most accelerate freshwater species recovery over the next 5 years? Related to your project, of course, maybe.
If we're so for Europe, if we stay with the targets of the Water Framework Directive and the implementation of the good ecological status by 2027, if we would really implement these goals, that would really accelerate the restoration of freshwater for sure within Europe. Because it's already on a political level, or it would be on the political level that we actually need.
Yeah, that's a very important issue for species like sturgeons, no? Anybody else?
Yeah, I
Jeremy.
think for many endemic species, after we identified the important threats and big problem in the freshwater ecosystem, I think creating the habitat and in situ conservation site that free from the especially invasive species and provide habitat protection in
Oh, I think his image is froze. Could you answer, Jeremy? We get Dicky back.
Yeah, sure. And really building on, I think, what where Dickie was going with his comment is the role of data-driven conservation action. It's so easy to go out and just throw the whole kitchen sink at a problem, but I think the more we can understand the threats and the needs of these species, the more impactful we can be. So I think from our corner of Africa, two things really stand out when we're looking at this, and that's being able to really identify what are the low-hanging fruit that can be implemented relatively cheaply and quickly to assist some of the most threatened species, and that's something we're really trying to focus on, almost taking that SHOAL list and getting a little bit more granular for the different regions where these species are concentrated to figure out like a really achievable and impactful sort of sub roadmap for the region. And just the other big thing, the other ingredient which I think is critical is the partnerships. I mean, we've got a big capacity shortage to do the interventions that are needed to recover our most threatened species. We have lots of critically endangered species that are not being assisted, and I think building that capacity through basically bringing different organizations on board, and importantly coordinating among the different organizations working in a region is a really great way to scale up the impact to all the species that need it most. Thanks.
Yeah, thank you. Michael. You want to say something, Mike?
Yeah, one of the things I mentioned in my presentation was there's the strategic pillar about inspire, and people like Jeremy takes these amazing photographs of fish and environments of people in freshwater habitats. It's so critical to this to engage a much wider audience. So the partners I was also thinking are very important. There's many people across the globe that keep fishes in the aquarium or some of the Cardina that Dickie's working on at home in their aquarium. They're massive supporters of or let's say have a love for fishes, and you've got organisations like zoos and aquaria that have the ability to sort of reach out to completely new communities. I think that building that partnership is fundamental to the success of conservation for freshwater species globally, because and Thomas talked about the sturgeons being a sexy species. We need so much more of that, because otherwise it's just a spiral where people aren't hearing about these issues, don't care about these issues, so no one's funding it, and so no one's got the funding to do the action. So building up this care and this community that care about freshwater species and aware of how amazing, incredible, and threatened they are is really is an important part of what we all have to do, I think.
Yeah, you're right, and Jeremy was mentioning those low-hanging fruits, because we need success stories. No, we need to show that we can make a difference, and yeah, that's very important. So I'm going to turn to some questions from our audience. I have one here for Jeremy. Ikram from Indonesia wants to know how to make sure that rotenone does not kill endemic fish and not only the invasives in the river.
Yeah, good question, and it comes from the right place, because rotenone targets all gill breathers, so it's indiscriminate. It's not species specific, really. And what we've got in South Africa is this situation where the invasive fish have done a very good job at displacing the indigenous species. There are usually very few, if any, indigenous species left after those bass and those bluegill and others have been in the river for a few decades. So what we do before we do the treatment is we do a native fish rescue, but the collateral damage is very, very small, because there's so few left in habitats where we're rehabilitating.
Yeah, the use of rotenone has always some questions like that. So I have another question here from Phil Boone from the UK. This is for everybody. What role do protected areas have in advancing freshwater conservation, and how can we deal with the potential need to alter the boundaries of protected areas as climate change affects species distributions? Complex question from Phil. Anybody wants to answer that? Mike?
Yeah, I mean, I have a go. Obviously, protected areas are critical, and there has, but very often they're not designed for freshwater habitats, or they're not or the freshwater habitat, like a river, is used as the boundary of the protected area, and it's not really considered part of the management plan of the protected areas. So ultimately, protected areas have absolutely very, very important role in freshwater species conservation, from the tiniest small protected area to the largest sort of landscape scale protected areas. So that, as I mentioned before, very often the protected areas may not even know they have important freshwater species in their protected area. So building that awareness, but also we have a big opportunity with the sort of rise of OECMs, other effective conservation measures, and I think this is particularly an opportunity for freshwater species, where rather than creating a sort of a strict protected area, you're able to create the conditions for protection of those species within the communities or within the habitats that people are constantly using, or finding different measures and mechanisms to protect them. So the answer to me is that protected areas are absolutely critical, and more of those protected areas can get engaged and understand the freshwater species that they have in their protected areas, the better. But also there's a huge opportunity to create new protected areas for many of these species that need protection, such as the place in Indonesia where there's only 15 hectares of the habitat left for betta burdigala that we need to protect, but also the opportunity that OECNs has and could be for freshwater species.
Oh, thank you very much, Michael. There's a question for Thomas. I wonder what is the opinion of the sturgeon community on the major name change as Spencer to Sterletus. What do you think about that, Thomas?
So there's actually just a paper in review that argues strongly for keeping the old names, as the new name changes are not based on a very valid phylogeny. But it would go into too much detail to tackle this right now. But overall, the sturgeon scientific community is not happy with the name changes.
Okay, thank you, Thomas. This is very interesting from Sarah from WWT. I'd love to hear your thoughts on what role conservation social science can play in supporting freshwater fish conservation across the world. Consider their importance for culture and livelihoods. How can we incorporate more social science into projects like we have heard from today? Who wants to talk about this? Dicky.
Yeah, well, yeah, I think it's very important to accumulate the expert for the conservation social sciences, because what happened in the many local NGO, especially in Sulawesi, we usually comprise our members usually come from the biologists, but we lack of support from the experts that have experience with social science, especially working with the conservation, because when we do conservation in freshwater endemic species, we also need to pay attention to the social and economical aspect, because just like the invasive species in here, we try to eradicate the invasive species, but at the same time, the invasive species also have several economically important for the local community. So we need to sit together with the various stakeholders, including the government and the experts from the conservation social science, to how to develop the best way to invasive for the non-native species, to introduce them into the freshwater ecosystem, but how to also to mitigate that they cannot have a big impact to the endemic species. Yeah.
Thank you very much. Well, we ran out of time. Thank you very much for your participation. We've heard some really important projects related to generating information, education, communication, ex situ management, and I think it's great to have all these projects going on in the world. And I thank you very much for all the work you're doing. I'm gonna send the microphone to Ana Nieto so she can give us our closing remarks. Ana?
Thank you, Topis, and thank you, everyone. I think it has been a very insightful session, and I hope you find inspiration in the work and experiences shared by all of you. I mean, for me, I'd like to perhaps just highlight a couple of messages. I think that the situation for our freshwater species is critical. I think we need to scale conservation action. I mean, I totally acknowledge the great results that our small grants have achieved, but I think it's also time to really scale conservation action for those kind of actions that require bigger interventions like habitat restorations and the like. And also, just to echo a little bit what also the other speakers have said, that freshwater conservation is only possible through collaboration. So whether through river restoration, community partnerships, or scientific innovation, each contribution plays an essential role in safeguarding species and ecosystems that are vital for both people and nature. So I'm also glad that this webinar has facilitated certain connections. I've seen in the chat many of you being in touch. So this was one of the objectives of organizing this session. So I'm very pleased with seeing this happening. And I just want to thank you, all of you, Mike, Thomas, Dicky, Jeremy, and you, Topis, for the facilitation. I want to thank especially our donors, Fondation Segré and the Save Our Species Sturgeons Initiative, for the continued support and engagement. And I just want to leave it there. Thank you all for the participation. I hope to see you in the next webinar, and keep well. Thanks, everyone.
Thank you. Thank you, everybody.
Thank you.
Bye-bye.
Everyone.
Bye.
Thanks. Bye.
Bye.
Bye. Nice to see you.